So it's a real pleasure for me to talk to you today. We'll discuss the genetics of autism spectrum disorder. And I just wanted to give you a very warm welcome from Houston, Texas, from Texas Children's Hospital and Baylor College of Medicine, which have provided excellence in medical care for individuals with autism and their families for many years, and who've also been at the forefront of autism research. Many autism genes have been described here over the past 20 years, and we have a large number of ongoing studies to better characterize and describe individuals with autism spectrum disorder, but also clinical trials testing out new therapeutic paradigms for individuals with specific types of autism spectrum disorder. There are a couple of objectives that are mentioned and that are also in your handouts. I hope that by the end of this presentation you'll be able to discuss the involvement of a medical geneticist in the care of individuals with autism, that you will be able to identify those individuals with autism who should undergo genetic testing. I hope that you would be able to describe first and second tier genetic tests in the evaluation of autism, and that you yourself are comfortable explaining the benefit of genetic testing to patients and their families. Now, just briefly, a general background about autism spectrum disorder. This is a neurodevelopmental disorder or range of disorders that are characterized by certain typical features as outlined in DSM-5 that was published in last year's May. So, in order to make a diagnosis of autism spectrum disorder, certain things have to be present. There have to be persistent deficits in social communication and social interaction, and there have to be restricted, repetitive patterns of behavior, interest, or activities. In addition, these symptoms need to be present in the early developmental period, and symptoms have to be severe enough to cause clinically significant impairments in social, occupational, and other important areas of current functioning. But autism spectrum disorder can manifest in many different ways, and there are different types and symptoms. So physicians, psychologists, psychiatrists, and geneticists have tried to categorize autism and come up with certain specific categories, some of which I'll discuss here. We do talk about high-functioning versus low-functioning autism. That's mostly dependent on IQ and whether intellectual disability is part of the manifestation. We talk about essential autism, when autism is more or less the only clinical symptom. And we talk about complex autism, when autism is part of a more complex clinical picture that may involve seizures, congenital anomalies, and others. In particular, in genetics, we differentiate between syndromic and non-syndromic forms of autism, depending on whether the autism is part of an underlying, well-described genetic syndrome, such as 22q deletion syndrome or Phelan-McDermid syndrome or others. We also talk about dysmorphic versus non-dysmorphic autism, depending on whether dysmorphic facial features are present in the patient or not. Simplex cases of autism, that means that the affected individual is more or less the only person in the family who has features of autism spectrum disorder, versus familial, which means there are multiple affected family members. And then lastly, depending on clinical course, static versus regressive. So has there been a static clinical course, or has this child developed more or less normally for a while and then experienced an episode of developmental regression with the loss of certain developmental milestones? In addition, there are a lot of co-morbidities with autism, and one of the questions that comes up is whether those are thought to be etiologically independent, or whether the presence of certain comorbidities may be the key to specific autism subtypes. So one of the most common comorbidities is intellectual disability, which is actually seen in the majority of individuals with autism spectrum disorder. A lot of kids and adults with autism spectrum disorder manifest features of ADHD or anxiety. It appears that about 30% of all individuals with ASD have a history of epilepsy. And then there are other behavioral and psychiatric phenotypes such as depression, bipolar disorder, obsessive compulsive disorder, or oppositional defiant disorder. And as a geneticist, this is really one of the big questions, right? What causes autism? Is it in the genes, or is it the effect of environmental influences on the affected individual? And a lot of studies have looked into this, and some of the most conclusive ones come out of the 1980s. These are family studies, and in particular twin studies. So here we compare identical twins, which by definition have more or less all their genetic code in common. Their genetic code is thought to be identical to each other, versus fraternal twins, which are born at the same time, but genetically they're not more than siblings. So they share about 50% of their genetic code. Now, looking at identical twins, if one of the two has a diagnosis of autism spectrum disorder, the likelihood for the other one to also be on the autism spectrum is between 80 and 95%. We call that the concordance rate. And that is very different from fraternal twins. When you look at fraternal twins or dizygotic twins, if one of the two carries a diagnosis of autism spectrum disorder, the likelihood for the other one to be on the spectrum is only 5 to 20%. And in these studies, these numbers that I'm presenting to you are based on twins that grew up together in a family at the same time. So the environment is more or less the same, but it's really about the genetics that drives this difference. And you can do a formal calculation and you will get to these numbers. So when you think about the overall etiology, the cause of autism spectrum disorder, we think today that about 90% of the causes are genetic and only 10% of the causes are environmental. On the other hand, that does not mean that autism spectrum disorder is typically inherited, meaning that it's passed on from generation to generation within a family. The pedigree that I'm showing here is actually the most typical scenario, and that is that there's only one affected individual in the family. And, it can still be genetic based on the underlying mechanisms, some of which I'll discuss later in the talk. Over the past 10 to 20 years, we've learned a couple of lessons, and some of which I've already mentioned. So ASD has many different manifestations. The etiology of ASD is mostly genetic, but ASD is rarely inherited. We've also come to the point, and this has been a humbling experience, that the genetics of ASD is very complex. The list of genes that are implicated in autism spectrum disorder is ever growing, and we've come to a number of greater than 500 genes, mutations or alterations of which can predispose to autism spectrum disorder. And even the most commonly altered of these 500 genes only account for less than 1% of all cases. So that really means there's not a single gene that accounts for, let's say, 40-50% of all cases, and then there are many others that account for the rest. But even the most commonly mutated ones only account for a very small fraction of cases. So some people actually talk about autism being a snowflake disorder, which means, people talk about snowflakes, and all snowflakes have common properties, but when you look at them under the microscope, they all look different from each other. They have a very particular structure. And this may be true for autism as well. They are shared properties, clinical features that are common among all individuals with autism, but then when you look at them at high resolution, when you look at them on a molecular and genetic level, they may be quite distinct from each other. Now let's talk about the genetic diagnostic evaluation for individuals with autism and what we have to offer today. First of all, and this is a very important point, the diagnosis of autism spectrum disorder is a clinical one. The diagnosis is based on behavior and not genes. So that also means, who makes the diagnosis of autism spectrum disorder? That can be the general pediatrician. It's all based on observation and the report of certain features and behaviors. Parental reports can also be teacher reports. And then the experts who, would make a diagnosis of autism spectrum disorder whenever the pediatrician is in doubt, those could be psychiatrists, pediatric neurologists, developmental pediatricians, or clinical psychologists. But then once a diagnosis is made, every family should be recommended genetic counseling, and every family should be offered genetic testing. And this is not just my personal opinion, but this is a guideline, a professional guideline put out by the American College of Medical Genetics, the most recent version of which is of 2013. What is a genetics evaluation? I just want to talk a little bit about this because not all of you may be familiar with the profession of a medical geneticist, and not all of you may have access to medical geneticists at your site. And we get asked, many times, so as a geneticist, do you actually see patients? Are you even a physician? Yes, there's a residency program of medical genetics, and as a medical geneticist, we're trained. As physicians, we do see patients in clinic. But we have a particular focus, right? We do take a medical history and we do a physical examination, just like every other physician, but we focus on certain particular aspects. Because the question comes up, how much of what we see clinically is caused by genes versus environment, we need to look very carefully at the pregnancy and get a good pre- and perinatal history. We do take a very thorough developmental history because we need to learn about the developmental course, the ascertainment of developmental milestones, and whether there have been episodes of regression or not. And then, genetics is all about family, so family history really matters. We do take a pedigree. We draw at least a 3-generation pedigree for every patient that we see. And we need to know about history of miscarriages, birth defects, intellectual disabilities, psychiatric disease, and others. When it comes to physical examination, we do a physical examination from head to toe with certain peculiarities, I should say. So we do take a lot of measurements. We take a close look at facial features, looking at particular facial dysmorphisms. Skin can be very informative, and the exam may also include the examination of family members in order to tease out whether a certain feature is a familial trait or whether that's really something unique to the child that may be a clue to diagnosis. A lot of the cases that we see have seen a lot of other providers before they come to our attention, so a thorough chart review is very important. We then make use of specific tests, and that involves chromosome tests, biochemical studies, DNA tests, and imaging studies. And just like anyone else, we consult with our colleagues. Some of the disciplines that we work most closely with would involve cardiology, dermatology, and neurology, I would say. So when families come to see us, they have questions. And the most important questions are listed here. The main question always is, what is the diagnosis? What does my child have? What caused this? Was this something that I did during the pregnancy or during neonatal life of my child? Or is this something that my child was born with that I could not influence? What is going to happen in the future? Do you have any anticipatory guidance? Will my child grow out of this? Will this get better? Or will this be a lifelong condition? What is the treatment? Is there anything that we can do? And what is the chance for this condition to happen again in my family, in future pregnancies? And then, people sometimes ask, both patients but also physician colleagues, why is it even worth knowing? Why does it matter? I mean, the diagnosis of autism is there. How does it matter to define whether this is caused by a mutation in the SHANK3 gene versus something else? So first of all, just like I mentioned, oftentimes it is about getting an answer. And especially mothers who come with their children, to clinic, they always wonder whether this is something that they caused during the pregnancy. And sometimes this can be a big relief to find out the underlying genetic cause and kind of put a scientific basis behind it. Then the chance of recurrence for future pregnancies. So some families may decide whether they want to have another child or not, depending on their chance of recurrence. And then once you know what the underlying cause is, you could consider prenatal testing or even pre-implantation diagnosis if this was a severe condition. But finding a genetic diagnosis really empowers families because once you know what exactly it is, you can find support groups. And in this era of the World Wide Web and everyone being connected, there are a lot of family support groups out there, a lot of Facebook groups. There's really a Facebook group for every rare condition. And this can be very empowering to families to find others who have children with the same exact condition. Sometimes we can give better anticipatory guidance, kind of looking back in the literature and looking at the 100 or 200 cases that have been described to better kind of tell the families what to expect in the future. And rarely, making a genetic diagnosis also changes medical follow-up and screening for certain conditions, or may even come with specific treatment considerations. Where do you find a medical geneticist? This is a humbling map, I would say, because we realize that there are large parts of Texas where there are not many medical geneticists out there. You can see that there's really kind of a hub here around the Houston area. And I realize that for some of you, it may not be easy to refer the family to a medical geneticist close by. So let's discuss, like when to consult with a clinical geneticist in the care of an individual with ASD, and what you can do, and what would be left to the medical geneticist to do. So following the guidelines that are put out by the American College of Medical Genetics, this would be a perfect world scenario, and that says refer every individual diagnosed with ASD as soon as the diagnosis is made. And then the medical geneticist can help with counseling, testing, that means diagnostic testing, DNA testing, and guidance. But when accessibility is problematic, then you may want to decide, and I don't think there's any problem in having the pediatrician order the first tier testing yourself. And then you could still refer the more complex cases, those kids who appear syndromic, those kids who have regressed or are in a phase of regression, those should definitely be referred to a pediatric neurologist or a medical geneticist. But I do think there's a lot that you can do, and hopefully by the end of this presentation you feel more comfortable about what there is that you should do once a child is diagnosed with autism. So let's talk about the specific tests. There's a little genetics 101 here, so the types of genetic changes that we can detect. There are different levels of genetic information that we can look at. First, we can look at whole chromosome changes, for example, trisomy 21 that causes Down syndrome. Then there are little changes of chromosome pieces that you wouldn't be able to see under a microscope. And when there are little pieces missing, we would call them a deletion. When there are little extra pieces of chromosome material, we would call that a duplication. And then, if we zoom in even further, we would get to the base pair level, which means the single letters in the genome, and we would spell that out. So we would spell the actual genetic code and look for misspellings. We would call those point mutations. This is one of the tests that will be most important in the workup of individuals with autism spectrum disorder. It's called chromosomal microarray analysis, and it has been considered a first-tier test for every individual with autism, at least for the past 4 years. It's a fairly new technology that is able to analyze the chromosomes for a large number of genetic disorders. It's a single test, and it's based on a blood draw from which DNA is isolated, and then the chromosome microarray analysis analyzes several hundreds of thousands of regions on the chromosomes. It has much greater sensitivity than the older methods of chromosome analysis that I will show in a second. So these are the traditional cytogenetic tests. From the 1960s up until today, we had chromosome analysis, which was based on isolating chromosomes from a cell and then staining them and look at them under a microscope. But the resolution here is very limited. In the 1990s, a new test became available that was called FISH analysis, which stands for fluorescent in situ hybridization. And this test has much higher resolution, but you can only look at a couple of sites at any given time. So in order to do this test, you need to know very specifically what you're looking for. And then chromosome microarray analysis came about in the early 2000s. And it's basically a FISH test, but it's hundreds of thousands of FISH tests done at the same time. And this is just kind of the general concept of this test. I won't go into too much detail, but patient DNA is compared to the DNA of a control individual, and each one of them is labeled with fluorescent dyes, and then they mix together and they hybridized on a chip. And there are specific sequences that are unique to certain areas on the chromosomes on the chip. And the DNA of the patient and the control will both bind to this chip, and then the excess will be washed away, and what will be read by a computer scanner, by a laser scanner, is the intensity of the fluorescent signal. And depending on whether there's an equal number, an equal amount of chromosome material in the patient and the control, you would see a certain color, but when there's an excess of patient DNA for a particular region, the color of that signal would change, and that would indicate a duplication. And when there's a piece missing, then there would be an excess of the of the control DNA, and that would change the fluorescent color, and that would indicate a deletion. And that's done, in multiplex hundreds of thousands of times. So it's all a question of resolution. The old chromosome analysis only was able to get down to a level of changes that are 5 megabases or larger, which means 5 million base pairs of genetic code or larger. The FISH analysis was able to go much deeper, down to a level of 40 to 250,000 base pairs, but only single sites at a time. And then the CMA has an average resolution of about 5 kilobases, so 5,000 base pairs, which means it's at least 1,000 times higher resolution than the old chromosome analysis. This is just a comparison. If you compare the old chromosome analysis to the map of a map of the world, you can get some information from that, right? You would be able to say whether New Zealand is there or missing, or if there was an extra copy of Iceland. But you wouldn't be able to tell much about Houston or specific streets in Houston. Now, the new test, chromosome microarray analysis with 1,000-fold higher resolution, is as empowering as Google Maps is, right? You can now zoom in and you can get much more information. You would be able to tell whether there's an extra building of Texas Children's Hospital, where we're at right now. So chromosome microarray analysis is performed on blood. Results are usually available within 1 to 2 weeks. And it will identify clinically relevant causes of autism spectrum disorder in anywhere between 7 and 20% of all cases, depending on the study that was done and depending on the severity of the autism. In general, I would say there's a higher yield in cases of syndromic autism and more complex autism, and there's a lower yield in non-syndromic autism. So general points here: the more dysmorphic your child is, the more complex the case is medically, and the lower functioning the child is, the higher your chances of identifying a relevant abnormality by chromosome microarray analysis. I would like to talk about a handful of other genetic or metabolic tests which may be considered in the evaluation of autism spectrum disorder. So the first condition here is Fragile X. Fragile X testing is considered a first-tier test for every male individual with autism because it is the most common single gene disorder to cause autism spectrum disorder. So Fragile X syndrome is a common cause of intellectual disability and autism in males. There are a couple of specific features in individuals with Fragile X syndrome, but I just want to point out, sometimes people know about the macroorchidism, the enlarged testicles, that are frequently seen in males with Fragile X syndrome, but be aware that you will only see the macroorchidism after puberty. So when a child with autism comes to you for evaluation at the age of 4 or 5 years old, you would not see this. Another clinical pearl is when you take the family history of an individual in whom you think he might have Fragile X syndrome, do ask for family history of ataxia or tremors in family members in their 50s or 60s. That can be a mild manifestation depending on the particular mutation in those individuals. Other metabolic conditions to be considered. Metabolic studies may involve plasma amino acid analysis, urine organic acid analysis, acyl carnitine profile, lactate, and others. Those are not considered first tier, and from a cost-benefit perspective, those are really not supported in any given individual with autism spectrum disorder. But depending on clinical course and the history, you may still want to consider them. Now, if kids were born in Texas and have had expanded newborn screening, many of these disorders, would have been evaluated for. But if the child was born in another country where newborn screening is not available or very limited, you may want to consider some of these tests. Do consider a particular test, and that is a test for lysosomal storage disorders for a condition called Sanfilippo syndrome, which is mucopolysaccharidosis type 3, if the patient has coarse facial features, just like this kid, with some mid-face hypoplasia, kind of upturned nose, kind of wide mouth, and just kind of coarse looking. Sometimes they may have an enlargement of the liver at the time of presentation, but they may not. And behavioral changes, regression, and autism spectrum disorder may be the first manifestations of this progressive lysosomal storage disorder. P10 sequencing. P10 is a single gene. This may be considered in individuals with autism who are macrocephalic. So kids who have P10 mutations usually have a head circumference way above 2.5 standard deviations above the average. Do have a high suspicion for P10 mutations when males have freckles on the glands of their penis. That condition is called Banyan-Riley-Ruvalcaba syndrome, or whenever patients with autism have vascular malformations. Then other genes that may be considered, MACP2 is a gene that when there are severe mutations, this causes a disorder called Rett syndrome, which is a neurological condition mostly affecting girls with developmental regression, acquired microcephaly, and the loss of purposeful hand movements. But mild mutations of MACP2 may only cause autism in females or not much more. Do consider carnitine biosynthesis analysis in males with ASD and significant developmental regression. This is, again, an easy test that could be sent on blood or urine. And this is actually a condition that is at least partially treatable because these individuals would be deficient of carnitine in their blood and brain during certain episodes in life. And L-carnitine can easily be supplemented and may help increase these levels back into the normal range and stabilize them over time. And then there's a whole new genetic tool which only has been available for the past 2 to 3 years. That is whole exome sequencing or whole genome sequencing. This test looks at the actual genetic code, right? The chromosome microarray analysis looked at the chromosomes in high resolution, but didn't look for the genetic code letter by letter. It has been clinically available since November of 2011, and studies to this date suggest that whole exome sequencing may uncover the genetic causes of yet another 10 to 30% of all individuals with autism, which means when you combine chromosome microarray analysis and whole exome sequencing, you may get to a diagnostic rate of 40 to even 50% for individuals with autism spectrum disorder, which is really a game changer when compared to what has been available, let's say, 5 to 10 years ago. I just want to highlight this difference between chromosome microarray and exome sequencing because people really get confused about this, and some people think that the chromosome microarray already looked at the entire genome. Yes, it did, but it did look in another way. So I use the analogy of a library. When you think about the genetic code as a library in each cell of your body, we have 46 chromosomes in each cell. This would mean 46 shelves filled with books in this library. And the books are the genes, which are the instruction manuals that the body needs to make proteins. So the chromosome microarray analysis would provide you with an inventory of the library. It would check all the shelves, make sure there's the correct number of shelves, and then it would look at the books on the shelves to make sure that each book is in the right place and has been returned, and that there are no extra copies of books. But it would not open the books and, look at the actual text. This is what the whole exome or whole genome sequencing does. It would open the books and check for misspellings, and it would look at approximately 30 million letters of genetic code in one test. And it will check every single letter, not just once, but usually 20, 50, 100 times to be really sure about what is being detected. Some people may wonder what exome means versus genome. So exome refers to the exons, which are the coding parts of the genes. So these are the sequences in the genome that are translated into proteins ultimately. Whole exome sequencing is also performed on blood. Results are usually available within 3 to 5 months. So this is a very involved test that takes a long time for analysis. It will identify clinically relevant causes of ASD in 10 to 30% of all cases. And there are a couple of ethical considerations here. So this is one of the tests that you should think twice before you send it, and you need to talk to the family about the power of the test. Because it is very clear that none of us has a perfect genome, and we will find variants in every one, hundreds of variants. So there will be variants of uncertain significance, and there's the possibility of pre-symptomatic genetic testing for things that you weren't looking for. So you may have a child with autism spectrum disorder and find by exome sequencing that this child is a carrier of a mutation in the BRCA1 gene for breast cancer and have very high risk for breast cancer. So this needs to be discussed with the family. There are usually opt-in and opt-out options so that the family can discuss what they want to know. But, finding things that you hadn't been looking for can be helpful because it may change the screening for this individual, and it may prevent breast cancer in the long run, based on having had the information early on. You could also uncover non-paternity, and that needs to be part of the consenting process that may be uncovered. So I'm getting to a couple of overview slides here, just to bring home some important points. Genetic testing in ASD. Importantly, autism spectrum disorder is a clinical diagnosis based on behaviors and report, not on genes. So the diagnosis is made by the pediatrician or neurologist, psychiatrist, or psychologist, not by the geneticist. Once the diagnosis is made, genetic counseling should be offered to each patient and his or her family. Chromosome microarray analysis is considered a first-tier test for individuals with autism spectrum disorder, and this should be offered to every family with a new diagnosis of ASD. And chromosome microarray analysis is really an easy test that can be sent from each location in clinic. There are hundreds of labs around the nation who offer this. And I encourage you, if you do not have access to medical geneticists nearby, that you become familiar with this and that you start sending chromosome microarrays yourself. Fragile X testing is considered a first-tier diagnostic test in all males with autism spectrum disorder. And then there's a whole number of second-tier tests, some of which we discussed. So Fragile X testing in females, P10 sequencing if the child is macrocephalic. Do consider lysosomal storage disorders if the individual has coarse facial features or hepatomegaly. Consider metabolic testing, including plasma amino acid analysis, urine organic acid analysis, acylcarnitine profile, creatine, guanidino acetate, carnitine biosynthesis panel, depending on clinical presentation. A lot of metabolic disorders have some episode of regression with them. Some of them tend to be more complex. For example, disorders of creatine and guanidino acetate metabolism often have epilepsy as part of the clinical manifestations, etc. MECP2 sequencing may be considered in females with autism, and then whole exome sequencing is really a new test to be considered, and whole exome sequencing realistically will replace a lot of these other single gene tests like P10, MECP2 and others. Importantly, however, whole exome sequencing will not replace Fragile X testing because the mutation that causes Fragile X would not be detected by whole exome sequencing. So with that, I want to get back to my objectives before we get to questions and answers. These were my objectives, and just let's go through them one by one and carry home some points. I wanted you to be able to discuss the involvement of a medical geneticist in the care of individuals with autism. So genetic counseling should be offered to each patient and family once a clinical diagnosis of ASD is made. The medical geneticist can help with counseling, diagnostic workup, and management of individuals with ASD. My second objective, identify those individuals with autism who should undergo genetic testing. Genetic testing should really be offered to every patient and family with a new diagnosis of ASD. But genetic testing always has to be targeted towards the needs and expectations of the respective family. So it should be offered, but there may be families who don't want to have any genetic testing, and that's perfectly fine. It also has to be discussed whether insurance would cover the expense of the genetic testing. Most insurance companies, especially for children, will cover chromosome microarray analysis with an indication of autism spectrum disorder. And up until now, most insurance companies will also cover whole exome sequencing, but some of them may require a pre-authorization. Describe first and second tier genetic tests in the evaluation of autism. We did go over that. First tier tests are chromosome microarray analysis and Fragile X testing in males. And second tier tests are the other ones that I just discussed. Lastly, I hope that you would become comfortable explaining the benefits of genetic testing to patients and their families. And this really gets back to these questions that are questions that families have when they come to the clinical geneticist. So why would you involve the geneticist? Because you want them to help the family better answer these questions. What is the diagnosis? What is the cause? What is going to happen in the future? Is there treatment? And what are the chances for this condition to happen again in our family in the future? Here are just a couple of helpful links and phone numbers. Feel free to email me any questions, or if in the future you need help with any of your patients, feel free to send me an email. I put down the telephone numbers of the Autism Center and the Genetics Clinic here at Texas Children's Hospital. But if you need to find a medical geneticist near you, you can go to the website of the American Board of Medical Genetics. If you need to find a genetic counselor near you, you can go to the website of the National Society of Genetic Counselors. The teleconference has been made possible through Title V funding awarded by the Texas Department of Health, and we would just like to thank all the facilitators at the various host sites.